Hi, good morning one and all. Welcome to today 8th Biology online video class. Today especially we are going to discuss about the third chapter, Story of Microorganisms. Story of Microorganisms. Of course, already we completed two more concepts in the previous videos. That is, what is science? Cell, the basic unit of life. Cell, the basic unit of life. There we discovered about the cell by Robert Hooke. Actually, the cell was discovered by Robert Hooke by his own microscope. Next, we discussed about the plant cell and animal cell. So first of all, bark or oak plant bark is a basically dead cell. Next, the live cells were observed by Anton van Leeuwenhoek. Plant cell and animal cell and the diversity of cells we discussed about in the second chapter, cell, the basic unit of life. Next, coming to the third chapter, story of microorganisms. Means what is the story of microorganisms? It means micro in the sense it's a very minute or a very teeny particles which we cannot see with our normal eye, naked eye. Certainly there is a tool required to observe these kind of microorganisms. The tool is nothing but a microscope. So around nearly 350 to 400 years back, there is no idea about these microorganisms. Why? Because there is no tool like microscopes was invented by the scientists like Anton van Leeuwenhoek or Robert Hooke. So the story of microorganisms was began after the discovery of their own microscopes. So the story of microorganisms was began at the time of the discovery. After that, we began the story of microorganisms or microbial world. I mean that the microorganisms is a separate world. Actually, the living organisms, not only the plants and animals, in between the microorganisms are exist in the nature, but we cannot directly see the microorganisms, but we can feel that effect of good or bad to the entire universe. So, coming to the story of microorganisms, part 1 and part 2. So here we get that part 1 means there is a part 2. Actually, part 1 is a very simple topic which are related to observing the observation of different kinds of microorganisms groups. For example, obviously we have the 6 different groups of microorganisms are exist in the nature. Those are obviously, first one is bacteria. It is a very familiar word in the science students. So bacteria is a next fungi. Actually it is a plant, but it is not autotrophs. They have to depend on the other organisms like saprophytes. They obtain the energy, they derive the energy from the dead and decaying organic matter. So normal fungus, this is called as a fungi. Next, protozoa are the first zoons, first animals like amoeba, euglena, paramecium. These are called as single cell organisms. We cannot see with directly with our eye. So these protozoans are the animals, means single cell microorganisms like animals. Fungi is a saprophytic microorganism like plant part. Next one is algae. These are nothing but the green saplings, the green mat-like structures. These are called as algae like Chlamydomonas, Oedogonium, Cara, Volvox. So this is called as algae. Next, microarthropods. Some minute arthro means giant legs like insects or aphids. Next one is viruses. It is a very special kind of microorganisms. There is a specific characteristic feature of the viruses are these viruses are the special kind. Why? Because they are having the connecting link. It is a bridge between the non-life and life. The virus which lives in the living body or host, then they are having the life because when the virus enter into the living body, they have to feed on the cells and they have to multiply their number and they cause the effect by showing some symptoms, so that they are having the life inside of the living host. But whereas it is outside of the body, there is no life. So it is just like a particulate matter which is outside of the living body or a host cell. So that is why viruses are the bridge between the non-life and the life. Till now, at the beginning class, I described about the living and the non-living cells differences and plant cell, animal cell differences. Now coming to the first part describes about only the group of microorganisms and where it is located and how to find out and what is the source of this group of microorganisms. While coming to the part 2, it is explains about that the part 2 is explain about the useful and harmful to that microorganisms to the nature. How the microorganisms are useful to mankind, how they affect the adverse effects to the entire the mankind, especially at present time we are suffering a lot with the coronavirus. Entire the universe is suffering with virus. So one virus can damage the entire the world. So it is a highly very risky factor to control over the viruses when they are growing in a rapid manner. So in medical there is a well-known proverb is there, prevention is better than cure. First of all, we have to focus what is the source of these microorganisms. Then we have to overcome to the microorganisms their population. So first of all, let me come to this topic. So the tiny particulate structures is called as microorganisms, which we cannot see with the naked eye. Certainly we need a tool or aid to observe these microorganisms. The tool is obviously called microscopes. So microbiology, actually biology is having a life science. Biology means a life science, study of plants or animals. It is a basic science or a basic life science. But these microbiology, biotechnology and microchemistry and these are all the applied branches. Biotechnology, microbiology, biochemistry. So these are called as applied branches. So one of the branch of applied biology branch is called as microbiology. Biology means study of living organisms. Of course, they are minute. It is a separate world. We can see, we can observe, and we can study these microorganisms with the use of with the useful of these microscopes. Next, coming to the actually the invention of microscopes in order to that the first one is Robert Hooke in the era it was in the era of 1665. Robert Hooke discovered a single lens microscope. Already we discussed in the second chapter, cell the basic unit of life. Apart from that, Anthony van Leeuwenhoek. Anthony van Leeuwenhoek is actually he is also a British scientist, and actually he is he's a British merchant, fabric merchant, means who is purchased these cloths and all. So for find out the better quality, in order to that he invented his own single lens microscope. So he is called as Anthony van Leeuwenhoek. So this is the picture regards to Anthony van Leeuwenhoek. So his single lens microscope which can magnify nearly 300 times. For example, one rupee coin it can be appears to 300 rupees coins nearly. So that much image can be magnified by this single lens microscope who has invented himself. So coming to the introduction part of this chapter, actually in those days, I mean that nearly 350 to 400 years back, there is some surprisingly there is some amazing questions. It is a puzzle to the entire the mankind. While we are wake up the bed, we feel some more bad smell in our mouth. What is the reason? How the milk is turned into curd? So there is a puzzle. So such type of questions, there is no answer at the time of 350 to 400 years back. But after the discovery of these microscopes and observation of the cells and microorganisms, now we have answer to these questions. Yes, these are the some adversible effects and these are the useful effects of microorganisms. So curdling of milk obviously by the lactobacillus bacteria. Lacto means the sugar which is present in the milk is called as a lactose. So lactobacillus bacteria. Lactobacillus bacteria means rod-shaped bacteria. So the bacteria which is present in the lacto means sugar, lactose sugar which is present in the milk. So it is a help to the conversion of milk into curd. That means curdling of milk. Next, actually Anthony van Leeuwenhoek, who is considered as the father of microbiology because he invented his own single lens microscope in the year of 1674, and he observed that some of the animal cules like protozoans. he called that the protozoans like amoeba, euglena, paramecium, these are called as a animalcule. Cule means it's a baby, that means a small one. He called that protozoans. First he called the animalcules, later it is called as protozoans. So observation of bacteria and RBC like blood cells and protozoans by the Anton von Leeuwenhoek. So that he is considered as the father of microbiology. Next, in order to the discovery of the microscopes, Jacarius Janssen at the year of 1595, it was that the first microscope was invented by the Jacarius Janssen in 1595. It is a compound microscope. So, coming to the textbook, what is mean by microorganism? What is mean by microbiology? So, microorganisms means the minute organisms which we cannot see with the naked eye is called as microorganisms. They are given that these microorganisms into groups. See, this is the first called as a cocci bacteria. Based on the shape of the bacteria is having a different names. For example, spherical shaped or oval shaped structure is called as a cocci bacteria. This is called as a rod shaped bacteria is called as a bacillus bacteria. This is the bacteria which converts the curdling of milk is called as a lactobacillus bacteria. Of course, we are having some permanent slides in our science lab that we are going to collect, and we have to observe these slides under the microscope. Then we can compare with the permanent slide while we are observing under the microscope and the picture given into the textbook. There is a comparison. Next, this is called as a chlamydomonas. It is called as a algae. This is called as a spirogyra. It is called as a ribbon shaped structures. These are the diatoms which are usually floating on the sea water, which is the feed of the sea organisms. It is the best seafood for sea persons. Next one is spirillum. It is a spiral shaped algae. This is called as a eudogonium. This is called as a seriaceum. So these are all called as algae. These are called as bacteria. So till now we discuss about that the groups of bacteria and the types of bacteria based on the shapes, and particularly about that algae. We are having the algae which is the rich source of that sea water or fresh water. Next, coming to that, this is called as a penicillium. Actually, the penicillin is the first antibiotic discovered by this Penicillium notatum fungus. Next one is the bread mold fungus or rhizophores, which is grown usually in the black molds which are appear on the bread. Next one, this is called as a mucus. Next, this is called as a aspergillus. These are all considered as the fungi. Next, coming to the two, these are called as amoeba, paramecium and verticella. So this is called as a amoeba. It is a protozoan. It is a fresh water free living organism, but it is a single cell organism which can perform all the activities within the cell. Next, this is called as a paramecium. It is a slipper shaped animalcule, but it is a shapeless organism. This is a bell shaped animalcule. This is called as a verticella. These are all under the category of protozoans. The first zoans means animals. Next, coming to the two, these are the microarthropods. Microarthropods means cyclops, daphnia and scabies mite, which are causing the scabies and eyelash mite. So these are causing to the two itching. So till now we discuss about that the groups of microorganisms picture. Now we are going to discuss about each one by one in a detailed manner. So on or before going to describe and a discussion about these microorganisms one on one, we are going to observe these microorganisms. We are having some permanent slides are available. So we are going to observe these permanent slides under the microscope. Thank you, children. Come to the science lab and we can observe the permanent slides. See, children, we are having some slides in our permanent slides box. So while we are collecting some of the slides which are related to our concerned topic, microorganisms. Actually, in the previous topic we discussed about that animal cell. So this is called as a permanent slide of animal cell. This is called as a permanent slide of plant cell. How to make the permanent slide? We have an idea about to make the permanent slide. Take a glass slide, put a drop of water and keep the specimen, and we have to add one drop of LESS0N and cover with the cover slip, and we have to make it a permanent slide. So this is called as a plant cell. So plant cell and animal cell, already we explained about the first chapter cell its basic unit of the life. Next we are going to rhizopus. This we call as the bread mold fungus. Rhizopus means bread mold fungus. We are going to now observe under the microscope. Next this is called as a spirogyra. Spirogyra means the ribbon shaped structure. This is called as the ribbon shaped microorganism called as spirogyra. This is called as a algae. Next one is these are called as bacteria bacillus, rod shaped bacteria. This is called as a bacillus bacteria, rod shaped bacteria. This is called as a lactobacillus bacteria, the curdling of milk. When we add the 1 or 2 drops of buttermilk to that milk, lukewarm milk, some moderate warm milk, immediately within a few hours the milk turn into curd. This is called as a curdling of milk with effect of these lactobacillus bacteria. So till now we discuss about the bacteria means lactobacillus bacteria and bacteria. Some of the fungus like rhizopus, the bread mold fungus, and plant cell and animal cell. Next to spirogyra like algae. And apart from that we are having some of the slides are available to the amoeba. It is a single cell organism. It is called as a protozoan. Amoeba is a single cell organism, unicellular organism like protozoan. Next this is called as a euglena. So amoeba, euglena, and this is called as a paramecium. So paramecium is called as a slipper shaped animalcule. So these are the slides which are related to observing the some of the microorganisms. Why? Because these are very minute in structures which we cannot see with our normal eye. Certainly we need a tool to observe these microorganisms. That is nothing but a microscope. So microscope is essential to observe this structure. So while we are observing the one by one, we can see this structure and we can find out the differences between the diagram which are given to the textbook and while we are observing the slides under a compound microscope. See children, just we are placing the pyramid slide under the microscope and just focusing the light. Just we have to adjust the lens according to the light availability. Then we can see the clear picture which are closely resembles to the picture which are closely resemble to the textbook diagram. See children, very good, excellent. So this is the picture which is clear to the closely to the textbook diagram. See, this is called as a amoeba. So like this one by one slide we are going to observe and we can find out the difference. Right, the recording the observations what we have to observe the diagrams. Thank you children. We will continue the topic next video.